What if you could write code inside a living cell, and that cell would run your program forever? Every living cell already runs on instructions. Those instructions are written in DNA, and they tell the cell what proteins to make, when to grow, and when to stop. Synthetic biologists figured out that DNA instructions follow a kind of logic, similar to the logic inside a computer. If this molecule is present, turn that gene on. If it's gone, turn it off. So they started writing new instructions from scratch, or borrowing pieces from other organisms and dropping them into cells. The cells read the new code and behaved differently. The basic building block is called a genetic circuit. It usually has 3 parts: a sensor that detects something, a processor that makes a decision, and an output that does something in response. One early example was a simple toggle switch built inside bacteria. Scientists could flip the switch with a chemical signal, turning a gene on or off almost like flipping a light switch. That sounds simple, but it proved something important. You can engineer reliable, predictable behavior inside something as messy and complicated as a living cell. One of the most talked about uses is medicine. Researchers are designing cells that can patrol the body, sense signs of disease, and release a drug only at the exact spot where it's needed. There are also efforts to program bacteria to live in the gut and detect inflammation, then produce a calming molecule right there, without the rest of the body being affected. Outside medicine, engineered yeast and bacteria are being used as tiny factories. You feed them sugar, and they produce useful chemicals, materials, or even medicines that would otherwise take expensive industrial processes to make. One real-world example is artemisinin, a malaria drug that used to come only from a plant. Scientists reprogrammed yeast to produce it, which helped bring the cost down and made it more available. There are also environmental applications. Some research groups are working on bacteria that can detect pollutants in soil or water and signal when contamination is present. The field is still young, and a lot of what works in a lab dish doesn't behave the same way inside a complex living system. Cells are not computers, and biology is unpredictable in ways code is not. Scientists are also thinking carefully about safety and ethics, asking questions like, what happens if an engineered organism escapes into the wild? And how do you make sure it can't spread in ways you didn't intend? The core idea, though, is genuinely new. Life as something you can design, not just observe. And that changes what kinds of problems biology might be able to solve. If you want to understand more about how the line between biology and engineering is blurring, there's a lot more to explore here. What part of this would you want to go deeper on?